we are doing this thing. Okay. Hello, I'm Isaac Larkin. This is Chris Norris. We are at BioBlaze, the DIY lab in Chicagoland area, and we are about to try our first nanopore sequencing experiment at this lab. I have a little bit of experience in nanopore sequencing from my job as a grad student at Northwestern, but we're going to be doing the rapid prep, and I've never done that one before, and hopefully it'll work fairly quickly because we actually need to get out of here in like an hour. So for those of you who don't know, this is a MinION DNA. It fits in the palm of your hand, costs $1,000, and it can sequence the heck out of a lot of DNA. And the way it does that is by threading the DNA through these little protein pores and reading the voltage as the different sequences go through. And from that, it can back calculate and call the bases of the DNA. So we are going to, for starters, just run the control sequence that Oxford Nanopore sends with its samples. And so that's just going to be the genome of this bacterial virus called the lambda-phage, which you can grow up a bunch of it and get tons of DNA from it. And so it's a good control to start with. Later on, we're going to see if we can sequence, like later on is in like in, you know, a few days or a week or two, we're going to see if we can sequence some plasmids from the Free Genes project, which we're involved with, and hopefully democratize sequence plasmids that people make. So we've got the software to run the MinION installed on Chris's computer. It's called Mino. We have the flow cell kit right here for the we have the rapid DNA prep kit. It's got the lambda DNA. It's got the fragmentation, which is a transposase that'll basically cut up the DNA and simultaneously attach a little adapter sequence to it that allows it to go through the rest of the sequencing processing and a few other buffers and things. This whole process should take like 10 minutes. So hopefully it actually does only take 10 minutes and goes smoothly. All right, so for library prep, I'm going to grab some. We may have the right tips. Let's see. Just wait, no, come on. What is that's? These tips are very okay. Oh yeah.
All right, solid.
We got the tips. All right. Now we just need a tube. All right, we found the PCR tubes. And the computer has to turn back on.
Yeah, if you edit this down a little bit.
Maybe we might edit it a little bit. Okay, cool. Got the tubes. Not mean to break that open as enthusiastically as I did. Good energy. All right. So we've got ourselves a thin-walled PCR tube. I believe we only need the one. I have two out just in case. Let me see if this thing's closed. Okay, cool. Now, one thing I'm noticing is that does actually take a remarkable amount of time to ramp up to heat. So what we may actually want to do the thing where, let's see. Okay. Hello, new viewers. We have the materials actually prep, or the, we have the materials and the tubes fully prepped now. And the pipette tips that we need. And so, we will actually begin the experiment. So we're going to mix 7.5 microliters of the lambda DNA, which is right here in this yellow tube, with, let's do that first. This tip touched my arm. I'm just gonna, don't wanna screw it up. Some nuclease is on my arm or whatever. All right, so I take 7.5 microliters of the lambda DNA right here, put it in the tube. Go. Then we're going to, we're going to use a different tip. Good practices. Oh, thanks. Yes, always valuable. There we go. And then we're going to take 2.5 microliters of the. Okay, so I got one thing I should do actually before I do that. Always important to make sure your reagents are well mixed. Otherwise, you might not actually get out of it what you think. How did that happen? What? Come on now. All right, that you didn't see that. Jesus Christ. All right. So, gotta mix the reagents really well. You know, gotta mix up those pipette tips, scatter them all over the floor. It's good science practices. All right, gonna mix up the lambda-phage DNA a little bit. Hopefully we don't shear it too much when we do that, because that would suck. So we want longer fragments. And then we're going to actually add it. Okay. 5 microliters of lambda-phage DNA. Well mixed. Going in the tube. Now, tip waste. Now we're going to take, actually, we're going to well mix the fragmentation batch. And the fragmentation batch contains an double-stranded DNA, and break it apart, and on the ends of it, place a little adapter DNA molecule that will be used in the downstream nanopore prep. Okay, that's mixed. Now we're going to take 2.5 microliters of it. And we're going to do this old school. So after we mix these two together, going to incubate them at 30 degrees Celsius for a minute. Actually, do you have a timer?
Yes, I do have it prepped.
Excellent. And once we've incubated at 30 degrees C for a minute, the fragmentation will have proceeded. And once we do that, we want to kill the transposase, the fragmenting enzyme, because if we let it go on, it'll fragment everything to hell, and the stuff will wind up with very short pieces of DNA. And the beauty of Nanopore is that you can sequence long pieces of DNA, and that makes assembly much easier. All right, so we're going to mix these two together. And now we're going to incubate for 30 C for a minute. All right, count it off.
All righty, counted. All right, you're good.
All right, incubated at 30 C for a minute. Now we're going to kill the enzyme. We're going to incubate at 80 degrees C for a minute. Cool. Let's let that run. And now we just got to cool it down a little bit. Let's just.
I don't know if we have ice, but I can get some cold water.
Yeah, we can. We got a little bit of. We got some cold stuff in here. Chill back. Just rub it on the chill pack. Do it. You know, this is how all science is done. Rub the tube on the chill pack. cool it down for a little bit. We're going to add the rapid adapter mix, 1 microliter of it. We'll mix it gently, flicking the tubes. Spin the tube down. We're not going to spin the tube down. We're going to shake the tube down.
All right.
Those arm muscles going. All right, that's been cooled down enough. Now, I'll actually just leave it on the ice. Why not? Oh no, we got to add the rapid adapter. I forgot. Okay. Now we're going to add the rapid adapter. 1 microliter. I'm going to grab a tip. One that did not fall on the floor. How did that happen?
It's nice to see someone of your caliber do something like that.
Yes, it should be very reassuring to you. Don't feel bad about anything. All right. Mix the rapid adapter well. Okay. Let's take a microliter of it. There we go. No bubbles in the thing. So it's about a microliter. And add it to the tube. Mix by flicking. The reason we're mixing by flicking is because at this point we don't want to shear the DNA. So, and pipetting too much can shear the DNA. So just flick it a bunch and that'll mix it up. And then once we're done flicking it, then we'll spin it down, get it all to the bottom of the tube, and put it on ice. Let's just slip it down there on ice. All right, and now we're going to prep the flow cells. So we've got our sequencing buffer thawed out, loading beads, and we've got the flush tether. Where's the flush tether? That's the flush buffer. Where's the flush tether? This is sequencing buffer. That's loading beads. That's sequencing tether. Oh, we don't need the sequencing tether, right? That's the thing we don't need. Yes, sequencing tether is not needed. We need the sequencing buffer. Flush buffer, the loading bead. Sequencing buffer. Where did they put the flush tether? I think that's over here. Flush tether. Aha. All right, we've got the flush tether as well. So basically, what we're doing with all this stuff is when the way Oxford Nanopore's sequencing works is you have to thread the DNA molecule through the protein pore, like I said before, but it has to be threaded through at a particular speed, and they have to be able to control that speed. If it threads too quickly, the signal passes before they can really collect it, or the signal becomes coupled to like the noise of like the sequencer being bumped around. So it actually needs to slow down the speed at which the DNA threads through. And they do that with basically a protein motor, a helicase that basically strips the double-stranded DNA into single-stranded DNA and threads a single strand through the pore at a set speed that's determined by the composition of the sequencing buffer that they have. Basically, like they feed the motor ATP and they give it sort of other stuff to make it happy, and the motor will run really quickly, and you'll get like 450 bases of DNA sequenced through the pore per second. And adapters that are put on in order to facilitate that process. Basically, one to stick the motor onto the ends of the double-stranded DNA, and then another adapter helps pull that whole complex down onto the little soap bubble, the little lipid bilayer membrane that the protein pore is sitting in, and then it'll sit around, be close to the thing, and then it'll get pulled in the pore and run. So that's what we're doing. And in order to do that, we need to mix the sequencing buffer And the flush, yes, by vortexing, spin down, and store on ice. I will say we aren't storing these on ice, and we probably should be for future experiments. But you know, it's the first experiment. They're fresh. It'll be fine. Open the lid. Oh, actually, one thing we should do is just jump to run. Well, it seems like it doesn't actually have a. Yeah, we should. Okay, we should check the hardware. Check the hardware. So we're just doing a quick test, checking the hardware, making sure the MinION is up to snuff, actually doing its MinION things the way it should be doing them. Always important. And we'll be doing similar things for the, once we've run this test, then we'll run a second test. The actual flow cell. So here we have MinION flow cell. That's packaging. Show you what it looks like. It's very, very well packaged. Always got to protect it. Oh, come on. There we go. All right. So MinION flow cell has a few different components. It's got the actual nanopore array, which is the stuff in yellow here. And I believe it has up to 1,000 pores for the MinION cells, different numbers of pores. And the numbers of pores affects how much DNA can thread through and be processed at a given time, and how much total sequencing yield you can do on a flow cell. So you've got the actual pores right there, and you've got this spot on port, which is where you insert your sample. Before you do all that, you have to prime the flow cell, because the buffer that the flow cell comes in is not the same buffer that you'll actually run your sequencing reaction in. And so what you want to do is basically turn this thing back here, and then you can, we'll do this priming reaction to basically flush out the storage buffer, and then the storage buffer will get pushed out and get stored in these little sneaky things over here. And then once we've done that, and we've prepped the flow cell with the sequencing buffer and given a little bit of time to equilibrate, then we'll actually add our sample. But first, we've got to check to see if we actually have active pores. So, done the check hardware. Seems to have worked, I think. I think. Let's see. Let's install that tonight. Whatever. Okay. Now we're updating. Updates. Is this it? Where's the thing? There we go. Messages. Hardware check has been successfully completed. All right. Our MinION is not a dud. That is very important and very good. So now we're going to take out the configuration test cell. There we go. I'm going to take out the configuration test cell, and we're going to put in the actual flow cell. This is the correct. Yeah. Just gotta push that in. It's a little bit squirrely because the thing pushes down really hard, but you just got to make these pins connect in this way. And just remember that, like, the snaky bit is near the port or is near the power connector, and it's also got this round right here, so you can tell. This, put it there, and then you check your flow cells. Check the flow cells, and we're also going to just show you real quick what I'm doing on this other computer. I run the software. So we're going to jump to run. Oh, actually, okay, cool. It's got the name, got the flow cell type. Oh, which flow cell type do we have? Let's check. 6. So the flow cell type is right here.
Gotcha.
On the flow cell thing. This is a flow cell Rev D chip. Okay, cool. The Rev D. It's got the new ASIC on there. This means that it has higher yield and lower leakage. That's good. Okay, so it's the 106. Good. And now we're going to jump to run. Oh, no, we're not going to jump to run. Yes. So select the flow cell. Now we're going to check the flow cell. Check the flow cell. Going to start the test. All right. So we've started the test on the flow cell. And what's happening now is the storage buffer contains some like calibration oligonucleotides. And basically it's turning on and it's going to its optimal temperature for the flow cell, which I think is like 34 degrees C. And then it's going to run briefly. And there's those calibration DNA molecules that are in the storage buffer to check how many pores are active and able to sequence on the thing. And hopefully that shouldn't take more than like 5-10 minutes. It doesn't. I don't remember how long. It shouldn't take more than 5 or 10 minutes. That's the hope. That's the hope, because we are running a little bit short on time. Now we just hang out for a little while.
Have more fun with the Opentrons.
Yeah, we could actually have more fun with the Opentrons. All right, I'm going to show you guys the Opentrons. It's just, oh no. I mean, the 0 people watching the thing, but just for posterity's sake, we have a lab robot, and we're going to use this lab robot to do great things. Interesting. Oh, there's the Raspberry Pi. Cool. It's just a robot liquid handler running on a Raspberry Pi from Opentrons. It was $5,500, $7,000, like with.
Yeah, I think 5,500.
The temp or the mag deck was.
The mag deck was like 1,500.
Yeah.
Shipping.
So we'll be doing fun things with this in the future. Hopefully building an open biotechnological foundry.
Hell yeah.
Built on a repository of free synthetic genetic parts. Should I be clicking jump to run? I don't actually. So MinION. So the MinO has been up the last used it. So it's not quite the same interface and process as it had before. It used to tell you when it was heating up the samples and when it got to the right temperature, or if it timed out. And it seems like it's not doing that anymore. So the question becomes whether, yeah, so this is the active run that we started. But is it actually started is my question. I feel like it should be actually started. Oh, hang on. Process. Yes. Okay. Our patience paid off. We didn't need to mess with it. All right. So it's processing MUX1. So the MUXes, I don't fully understand how they work, but they're multiplexers. So basically, there are 1,024 pores on here, but it doesn't run 1,024 pores at the same time. And the reasons it doesn't, I'm not actually fully clear on. It might be that like the limiting factor is not the number of pores you can fit on the device, but rather like the number of signal processing units you can fit on the ASIC. Maybe? I don't know. But in any case, there are more pores on the device than can run simultaneously. And so the first thing it does is it tests basically all of the pores on the device to see how well they're performing. And then it boxes them into like 4 different groups. And the best performing pores go into group 1, and they're the ones that get run at the beginning of this. So you get like most of your data out of that group. And then like when those start to peter out and start to break, you switch to MUX 2 and switch to MUX 3, MUX 4. Okay, so now it's processing MUX 2. That's good. 21% of the way done. All right, this is making me happy. Probably turn this off.
Same with the water bath?
Yeah, same with the water bath. I'll clean up the pipette chips as long as we're waiting. Just show what it looks like while running. Looks like you can see progress bar for the flow cell check running. Got the update messages over here. You can tab those like that, and then the very pretty MinION blinks red and blue and green when you run it. Or actually, I think whenever you plug it into the computer. It's a cool device. So cool. It's like honestly, like the, you know, when there's a lot of hype around nanotechnology, but the MinION is legitimately hardcore nanotechnology, like protein machines threading single molecules of DNA, and then, you know, semiconductor fabricated ASICs processing the signal from those single molecules at high throughput. Technology, man. All right, let's check in. Okay, cool. That's, our tube is still on ice. Very good. And we're way through. Good, good, good. Hey, look at that. Oh, wow. 1,516 pores. They have really improved since last year. So like last year I got flow cells and they had like 800 pores to start, and now there are twice as many pores to start. That's very cool. Rapid improvement. All right, we've got the thing, finished the platform QC. That means we are ready to prep the flow cell for the sample. Let's get, for this, you'll want a P1000. Cool. I'll crank the 1000 down to 200 microliters. You're not actually going to suck up stuff with this. You can actually use the ratchet, the dial on the pipette to do the sucking, because this next part is a little bit delicate. Because you need to do, here, just show the video. I don't, actually, could you hold this like?
Yeah, I got you.
Yeah, hold it so that you can see
right about here.
There we go. Yeah, so that you can actually see the device. All right. So what we're gonna do is we're gonna turn this dial back this way, okay? Expose a port, and that can put the buffer that gets the flow cell ready for sequencing. However, we want to be very careful to avoid air bubbles, because air bubbles will kill the pores. It will screw them up, and then you will have a dead flow cell, and each of these flow cells is worth like $900. So what you do is you take, in order to remove any leftover air that's in here before you start priming the whole thing, is you take your P1000 pipette, set to about 200 microliters. And then you don't pipette up, turn the thing until you get up to about 220. There we go. And you see you start to get a little bit of liquid in there. And actually, there we go. You start to get a little bit of liquid. And once you get a little bit of that yellow liquid, then you're in good shape because there aren't bubbles. And you can actually put a little bit, a little bit back. There we go. Good. And now that we've done that, we just need, actually, now I need the computer to, all right, because they changed these solutions. Okay, so now we're going to prepare the flow cell priming mix. We're going to add the flush tether directly to the tube of thawed and mixed flush buffer, and then we're going to mix it by pipetting up and down. All right, we can do that. We can do that here. Hold off this. Let's grab the flush buffer and the flush tether and mix them together. We've got the flush tether and we've got the flush buffer. All right, excellent. It's only 30 microliters. There's a lot more flush tether in here than 30 microliters. All right, mix it a little bit, spin it down manually. All right. It's definitely microliters of thawed and mixed flush tether directly into the tube of thawed and mixed flush buffer. And let me just make sure. I just want to make sure that I'm actually supposed to flush buffer. Yeah, it doesn't say anywhere to transfer X amount of flush buffer to a different tube. So we're actually, we're just going to add the flow, add the flush tether to the flush buffer. All right, so we're going to need a P100 for that. 100 right here, set it to 30 microliters. Grab some tips. Okay, we got tips over here. These will work. Just for liters. Let's well mix the flush tether. 2, 3, 4, 5, 6, 7, 8, 9, 10. That's well mixed. You know what? I'm gonna flush buffer that way as well. Because I remember at least one of these buffers. Like I got it a couple of Nanopore. They're like clearly something denser at the bottom of the tube, and it needed to get it moving up and around. So just to be on the safe side, we're gonna make sure this is mixed by pipette. Yeah, that's fine. Yep. Right, these are well mixed, and now we're going to add the 30 microliters of flush tether to the whole tube of flush buffer. And then we're going to do, 800 microliters of the priming mix into the flow cell by the, that's a lot of 30 microliters. I'm curious if the wash solutions come with like more of this, or what's going on there. All right. Now we're gonna mix it up, crank it up to 800. Wash it thoroughly. Good. All right. Get it well mixed. Some gentle pipetting. Good, good, good. Now I've got that 800 microliters right here. Or to the port. And just gonna make sure. Oh, blocks me out. Yep. from 800 microliters of the priming mix into avoiding the introduction of flow cell or of air bubbles. So it's going to go here. No air bubbles. All right. And we're going to push like that. You can see the yellow goes away.
Snaking its way on.
Snaking its way on through. All right. Good-ish. Good-ish. Good enough. There might be a little bit of storage buffer left over there. Hopefully that won't mess with our sequencing run too much. All right. Now that's going to sit for 5 minutes. Priming the thing. I'm going to set a 5-minute timer. All right, 5-minute timer. In the meantime, we'll prep our library with the loading beads. Well, 34 microliters of the sequencing buffer. Sequencing buffer. Mix this up as well. Plenty of it. 34 microliters of it. We'll want 25.5, yeah, I remember that, of the library loading beads. So the beads are basically these little, I think they're plastic beads. And they have modifications on them. I don't know the exact modifications, but they have modifications on them that allow them to bind to the DNA. And their job is to bind to the DNA and then fall via gravity onto the top of the flow cell where all the pores are. So it basically increases the local concentration of the DNA around the pores to increase the rate at which properly adapted DNA with a motor on it finds its way onto a pore and starts threading through. All right, so we're going to do 34 microliters of sequencing buffer, and we're going to do it in this tube. Best practice, I should label all these tubes, but we're only working with 2 different tubes, so my hope is that I won't mix them up. All right, tube number 2, 34 microliters of sequencing buffer. Go. Now, we want 25.5 microliters of the library loading beads. Library loading beads. We'll do 26 because this pipette doesn't do decimals. Which is, I mean, I highly doubt that it actually matters, being 25.5. And you want to pipette up and down a lot with these beads because they fall to the bottom. And what you want is a well-mixed suspension of the beads. So you pipette them up and down. And then you got your beads. Close that. You add those 25.5 microliters to the tube. Like so. And mix it up. You do this, you want to try to avoid making foam or making bubbles. It's okay to make bubbles, just make sure that you like shake the thing down or pop them or whatever. Bubbles are your enemy. All right, and then last component, type in my password yet again. Last components, all right, we want 4.5 microliters of nuclease. Did the thing come with that? I feel like
no, but we've got plenty.
Oh, you got plenty of nuclease for water?
Oh, it's in there.
It is?
Yeah.
Where's it? Oh, wow, yeah.
Yeah, we also
aliquots?
yes, yeah, we made a ton of actually. Okay, I'm gonna set this down really quick.
All right, that's 5 minutes. That means the flow cell is primed properly, which means that this 4.5 microliters of water that we'll add come at just about the right time to this tube. All right, 4.5 microliters. Good. There's the water. There we go. And finally, 11 microliters of our fragmented and adapted DNA library. Let's see if this is 11. Sometimes they do. Nope. All right, that's fine. That's fine. We'll use this one. 11 microliters. Actually, we can probably get away with this one. Just for the sake of precision. It probably doesn't matter because I think the entire library is 11 microliters. Yeah, the entire library is definitely 11 microliters. It doesn't actually matter. Okay. This whole thing, just transfer the entire contents of this tube that you've made.
Mm-hmm.
It contains the DNA and the fragmentation mix and then the rapid adapter mix. Transfer all of that. All of it. Every last bit of it. Come on. Get out of there, too. There we go. Every last bit of it into this tube. Good. And now going to mix well. Interesting. Okay, cool. Cool. Mix gently by pipetting up and down just before loading. We will do that. We will mix this gently by pipetting up and down right before loading. But first, the first thing we're going to do is finish priming. Because we primed with 800 microliters, but we need to prime the remaining 200 microliters of the priming mix. All right, so you've waited 5 minutes after adding the 800 microliters of priming mix, and now you take the remaining 200. Well, actually, a little more than 200 is left. That's fine. Good. They gave you a little bit of wiggle room. And you know what? Because they gave us a little bit of wiggle room, because I'm not entirely sure that it got all the storage buffer out, we're just going to crank it up to 250. Why not prime a little bit more? All right. Now, just stick it on there. Form a seal. Don't form bubbles. And gently pipette. Pat it all down. Flush it out. There we go. Good. And yeah, we've got that. And now we're going to add 75 microliters of sample onto the thing. So we're going to close this, I believe. Okay. Now the thing I don't remember for sure is whether we keep this open while spotting on here or whether we close it. So it's a decision to make. We're going to need to close this first. I think I need to close it first. I'm going to close it. Close this down. There you go. Open up the spot on port.
check to make sure people can see. Let's put it down like that. All right. Gonna open up the spot on port, and we're gonna see if we can actually show this how it's done. I'm getting a phone call. Where am I getting a phone call from? Probably getting a phone call from Alita. Nope. Oh, Delaware. Hello. Oh yeah, send him up. Yeah. All right, thanks.
Neat.
My wife's guitar instructor has arrived,
Oh yeah.
and yeah, for some reason they have our phone numbers mixed up on the thing, and so whenever someone arrives and asks for Lita, they call me, and whenever someone arrives and asks for Isaac, they call her. And we've never been able.
Yeah,
to
that
get
makes sense.
We've never been able to get it changed, and so we're just like, oh yeah, you know, that person's there. Yeah, that's for the other person. Sure. Probably shouldn't have said that on the internet. It'll be fine. Okay. Now we're going to add things to the spot on port. All right. Hopefully this is how it's supposed to be done with the priming port closed. If not, I mean, it's fine. We can always just open up the priming port. So we'll just be gentle and careful in our first drop. All right. This is our library. We're going to put it on very gently. And it's not going anywhere, which seems to imply to me that I, in fact, do need this open. Let's see. Open up and see if anything changes. There we go. And now let's add the thing drop by drop. There we go. Okay. Priming port open is the consensus. And what I'm finding here is that it's actually much easier to gently add by ratcheting down the pipette rather than actually squeezing on the pipette tip. So that's a little tip for you right there. Not okay. Hopefully this. Want the oh, now it's not going through. Come on now. Is there a plug? Is that what's happening here? There we go. If it gets stuck, one thing you can do that helps is you can all right. We've loaded the library. If it gets stuck, you can very gently form a seal on the hole, and then you can just ratchet up to suck up a little bit of liquid.
Gotcha.
And that'll unstick it without adding bubbles. All right, we've added our library. The time has come to run this thing. Yeah, let's do available. Let's do new experiment. And our experiment will be, let's name it 2018 1110 Lambda Rapid Control, Lambda Control Rapid. Rapid prep. Long name. The sample ID. Edit flow cell. I don't actually know what the sample ID should be. It's good that the flow cell ID is known to the thing now. That's nice. Upload a sample sheet. We don't have a sample sheet. We'll just call the sample 2018 1110 01. I don't know, something like that. hopefully that'll be fine. Okay, cool. No devices available to add. Good. Good. That's the experiment. Now we're going to set the kit. Our kit is DNA. It's PCR free. It's, I believe our kit is the RAD 0004. Let me just double check that. This is the flow cell washing kit. That's the flow cell priming kit. Where? Oh, here it is. All right. So our kit is SQK RAD 004. That's the rapid prep kit. Good. Base calling. Yes, we do want base calling. Run options. Ooh, we could set the length of the run. That's very cool.
I am all ears, Isaac.
It needs to be connected to the internet. Damn it. It needs to be connected. I was going to say, so you know what we can do?
What can we do?
We can start this run now, just to make sure it's working.
Gotcha.
I believe we can then pause the run. Then you can take all this stuff to the TED Talk, and while the TED Talk is going, as long as you have connection to the internet, you can restart the run.
Solid.
I believe. I think that'll work. So we're gonna say. I mean, so how, like, would you be comfortable leaving for the thing at like 11:20?
Yeah, that's fine.
Yeah. All right. Let's run it for 0.5 hours. 0.5 hours. And active channel selection on. Time between mux scans. Sure. Why not? Output. Yeah. Fast 5. Cool. This is what you want. So the fast 5 gives you the raw data, and this is where you find the base called read. FastQ is the output. Like once it's base called, it'll output to a FastQ file, and the FastQ files are smaller. We don't have any custom scripts, so now we're just going to run. And experimental parameters are complete. Close it up. Close it up. Let's see what's happening. Calibration finished successfully. Farther away from the edge of the table. Just, minimize the risks of the MinION falling on the floor. I'm certainly a clumsy person. All right. Put that there, put that there. Moving this around. Start cleaning up a little bit. Start putting stuff back in the freezer.
Sounds good. I'll set this thing down.
Yeah. Let's push this over here a little bit. Just a little bit. Nope. Is there something back there? All right. Just one opportunity to see what's going on. MinION down here. There we go. And experimental parameters complete. Because we'll want to show what we'll want to show what the software looks like when it's running, because that's useful to know for people, generally speaking. Okay. Let's start cleaning up in the meantime. All right, we got it running. We got it running.
You did it, man.
It took a little longer than 10 minutes, but it's all things that are good take a little bit of time.
Hear you there.
And hopefully what we'll find once it actually starts running is that hopefully what we won't find is that I've completely screwed up the library in some way and it has terrible yield or terrible size distribution or things like that. We'll see,
Worst comes to worst, it's still a nice trial run.
Yeah, it is a nice trial run. And you can try repeating it tomorrow.
Oh, I will. And boy, will I disappoint. But I will give
No. It's not true. Oh, okay, cool. So the flow cell priming kit. Oh, there's the stuff for the flow cell priming kit. Okay. Yes, they did come with plenty of flush buffer. Good. That's why they've got that flush flow cell priming kit. And then this last bit of flush buffer. I guess we don't need it. Throw that away. It's too small of an amount since I've got the flush tether added to it. All right, got all those. Put those back in the freezer. Okay, let's see. Is the thing running? Nope, still get set up. Like this. I'll set this in here. Oh, we haven't shown how to wash the flow cell. Washing the flow cell doesn't take a long time. We'll let it run a little bit and we'll yeah, then we'll wash. It'll be probably better to just run this for 30 minutes and then wash it and put the flow cell back in the fridge so that we maximize the flow cell's lifespan.
Gotcha.
Maximize the amount you can get before we need to plant another.
What's the price on each one of those?
So each of these individually is $900.
Oh wow, wow.
Yes, which is why the flongle is a big deal. So as soon as we can start getting it, as soon as we can get a pack of 50 flongles, we'll be much happier because those are only $100 each and get closer to the scale that we need to run.
Hear you there.
And hopefully we can figure out a way to run the flongles that allows us to use them multiple times. They're not supposed to be used multiple times, but I suspect that we could probably get around that. Mod the protocol.
What prevents us from using those right now?
They are so they've been released to developers. You could reach out to Oxford Nanopore and ask them if it's still possible for us to buy into the early developer set.
Can't hurt to ask, right?
Yeah, I mean, so it's like one thing to consider is the early developer set. They might have a few more lemons in them than the final consumer product. But you know, it's not like we need like massively high yields on them anyway. I mean, maybe for some applications we'll wind up like outsourcing Sanger sequencing. It's so lame. Sanger sequencing. You need a primer to do it. I mean, I guess if for the Free Genes project we're, okay, cool. Hey, look at that. Okay, so it's done a muck scan. Interesting. All right, so it found 1,233 wells this time. And now it's starting the sequencing. Okay, cool.
Solid.
I'm excited to see what the yields and stuff look like. But yeah, since we're gonna be like designing plasmids from scratch for the Free Genes project, we could probably build into all the plasmids like a universal Sanger primer site. And for like low throughput engineering projects, that might be valuable because like the per run cost of Sanger sequencing is like $7 to $10, I think, or at least it is at Northwestern. I don't know what the private rates are. And I mean, if the choice is between, if you're like only making like 2 or 3 constructs, then that makes more economic sense than the Flongle. Although even then, if your construct is longer than like 700 bases, like Sanger sequencing only does like maxes out at like 700 bases of sequence. And so if you need to sequence a longer thing, you actually have to have like another primer and then sequence more along it. And that's another Sanger run. And so that's another $7. So for most use cases, the Flongle MinION combo, especially for like several different especially for like 10 plus constructs simultaneously, especially for longer constructs, will be more important. Although actually, this reminds me, the thing that I was really thinking through is I think in the short to medium term, once we get the FreeGene MoClo assembly standard working, and so we'll basically have like this set of plasmids, and all the plasmids are sequence verified. And so you just cut out the parts from the plasmids and stitch them together. Like you're stitching together a bunch of sequence verified things. You don't actually need to do, it's not a huge deal. You don't really need to do sequencing on that. And even if you needed to do sequencing, or even if there was a chance there was some sort of mutation that might throw things off, once we get the cost of screening is going to be lower than the cost of sequencing. So you can just screen for function, and the ones, and you're going to be screening for function anyway, because like, with any given genetic circuit, you like design it and then it doesn't work. And then you're like, okay, why isn't this working? Let me go troubleshoot. Or you design like 5 of them with like, slightly different architectures or slightly different parts fitting in. One of them will work, and then you go back and be like, okay, why did this one work? I'll use this for my thing. And so if you're going to be screening a bunch of those anyway, and you're going to screen like a few replicates of each of the ones, and your cost of mini prepping and cloning and assembling and transforming is like on the order of 5 cents per mini prep, maybe like $1 per screen, then it just makes sense to you can do sequencing as an afterthought once you already have the device that works and you've characterized it, and finally you're like, okay, I have this thing that works. Now, I need to, verify that this is the actual sequence that it is, and so I will sequence it. So it says it's running. It says it's running, but my question is, where do I see that? Oh, here we go.
Solid.
Look at that. Okay, so something weird happened here. There are a bunch of dead pores right here. I'm not sure how that happened, but for the most part, that's not bad actually. Like that certainly looks better than my PCR amplicons that I ran a year ago. Look, something weird. I did something weird and it killed a bunch of pores right here. I'm not sure what that was, but it happens, and maybe hopefully we'll recover those at some point. But yeah, for the most part, like this is a lot of it's got 200 pores sequencing simultaneously. 6 are open, and yeah, we can start looking at. Look at that, yeah. Interesting. It's maybe not great that the proportion of inactive pores is slowly increasing. I'm not sure why that's happening. But then again, this is a short run anyway, so it's not like we're trying to get this thing to run continuously for 48 hours right now. All right, and now it's being a little more constant. The number of inactive pores is still slightly increasing.
What do you think, Scott? Isn't it?
I have no idea. Like, is there like a bubble happening right now? Let's see.
No, I don't see a bubble. I'm not sure. But if we go back up here, yeah, it's consistently right around here. This area on the ASIC muck scan results. All right, look at that. Ooh, that's a pretty curve. Look at that. Wow, we're already up past 7,000 reads. That's really good. That's cool. All right, so we're up past 7,000 reads, and of those reads, it looks like about 1,000 of them have been base called. Oh, now we're up past 8,000 reads. Just past 8,000 reads. Now let's see. Oh, what's the read link histogram look like? All right. It's not terrible. We've got a few reads that are really long. Like this read is 40,000 DNA bases. This read is 49,000 bases, and it looks like the main proportion is right in the range of 4.7, 3.9. Yeah. So this is 6.5 KB. So most of the reads are like 1 to 5 kilobases in length. It's good. Only a sort of relatively small fraction of them are shorter than like 1,000 bases. That's good. That's very good. Interesting. The bucket size. You can modulate the bucket size. Oh, that's cool. They really have upgraded Minnow since I last used this. That's nice. And what kind of quality scores are we getting? All right. I mean, these are actually pretty good quality scores for raw reads. So, this is sort of like how noisy the reads are. How noisy does the base caller think the reads are? So, and the way this is calculated is like it's like the negative log base 10 of the predicted error rate, something like that. So basically, like if you have a quality score, a Q score of 10, that means that read has a 90% accuracy rate or a 10% error rate. And it looks like the median for our reads is up above Q10, like something like Q11, and creeping a little bit into Q12. And that means that most of our reads have quality scores above 90%, error rates lower than 10%, which is what you want for a nanopore read. I mean, like if you do 2D reads, the first strand, and then you read the second strand, and there are a couple of prep kits they have for that, but you won't be using those because they don't have rapid prep kits for them. You can get up to like Q20, so like or you can get close to Q20, so close to a 1% error rate for the raw reads, which is pretty insane for like single molecule sequencing. You know, like even PacBio, which had single molecule sequencer that just got acquired by Illumina, they pride themselves on like the accuracy of their consensus, but they've been in business for like years, since like 2009 or something like that, and their raw error rate is still, if you sequence around this thing, 20 times, then your error rate is 15% of 15% or 15% times 20, so you get like super accurate. And with Nanopore, it's mostly random, but not entirely random, and so they still have to sort of figure out ways to get rid of that systematic error. But they're getting there. They're really I'm impressed. I'm very impressed by the algorithms, just like the algorithmic improvement they've done to get up above Q10 for most reads is that's very cool. Like I think last year most of my reads were below Q10, and now most of the raw reads are above Q10, and it's only going to get better because now they're doing the orthogonal pore thing that they're figuring out. And so like they can get consensus accuracy of like one error in between 10,000 and one error in 100,000. And so once they actually start selling the flow cells with like the two orthogonal pores, with the R10 pores, then I mean
at that point, I don't know why you'd buy an Illumina instrument, except maybe like per run cost on certain things. Honestly, we don't need more than like 50 megabases of this. So 1111, we have more than enough data to, because like the lambda phage genome is like 50 kilobases.
All right.
So right now we're at 1000x coverage on that.
Oh shit.
So we don't really need more than this, actually. We're pretty happy with how this is going. Let me just see how much time is left on the experiment. I think it would be like 20 minutes. Let's see. No, it's only 42%. Yeah, let's stop the run.
All right.
I wonder if we stop the run, do the non-base called reads keep getting base called? Who cares? Yes, I'm good. Okay, base calling will continue. That's excellent. That's very excellent. All right, cool. So we've stopped this run. We've successfully done our first MinION run. Now we're going to clean the flow cell so that you can do this again tomorrow.
Sounds great. I'm going to throw this to the side really quick.
Wash kit. Wash kit. All right. The wash kit requires wash solution A and storage buffer, I believe. So cool. We have it working. The number of inactive flow cells did continue to increase. I wonder what that's about. I don't know what that's about. It's annoying. But that does tell you, like, you get 50 megabases of data from this thing in 10 minutes of sequencing. So, like, you know, imagine the average plasmid is 5,000 bases long. You have 10 plasmids, 50 kilobase, 1000x coverage of that. That's, you could do like 100 plasmids and get 100x coverage on those plasmids in 10 minutes. And 100x coverage should be more than to get to like Q40, Q50 base calling accuracy on the consensus. So yes, this is much faster than Illumina. An Illumina run takes like all day or all night. That's awesome. All right. Now let's wash the thing.
Should probably get some more pipettes over here.
Huh? Oh yeah, for which? I believe I need the P1000. All right, success. Just gonna clean the flow cell now. Clean the flow cell. Okay. Protocols. Washing MinION flow cells. Yes. Consumables. Yep. That's all you need. All you need is a P1000 PET and tips and the flow cell wash kit. All right, we're going to open the priming port. Make sure the buffer is continuous. Always important. Make sure the spot on sample port is closed and the priming port is open. Oh, oops. I left the priming port open. But don't do that. Close the priming port afterwards. Okay. 150 mic. Oh, wow. Not actually that much. All right. All you need is 150 microliters of buffer solution A. All right.
All right.
So all you need to wash
Mhm.
the flow cells is the flow cell wash kit, a P1000 pipette, and tips.
Perfect.
And one thing that I forgot to do that is important to remember to do is once you have put on the sample and you're going to run the thing, close. Yeah, it's entirely possible that a bubble got in here, and that was why it killed a bunch of the pores.
Cool.
Um,
Oh, maybe cool is the wrong word.
but yeah, it shows you the danger of the bubbles. Solution A, making sure it's continuous. And the way you make sure it's continuous is you suck up a little bit of it, make sure there are no bubbles, and then you make sure the spot on port, the port where you loaded the stuff, you close that after you put the stuff on. And yeah, you only open that port when you okay, cool. Only open that port when you need to add the library, the DNA library, your sample. Okay, 150 microliters of solution A. Now we wait 10 minutes.
Perfect.
And then, no, we're not going to run a second library straight away. We're going to store the flow cell for later use. That's what we're going to do. I believe we literally, yeah, okay. Yeah, you just add 500 microliters as the storage buffer. Yep, that'll do it. Hi, Anoop. Nice to meet you. We're basically done at this point, but if you watch the rest of the video, you can see how to use a MinION DNA sequencer to sequence DNA. Yes, that's what we're up to.
Stop.
No, it's gotta wait. Clean up. And yeah, I'll try to record the results. Oh yeah, I should actually. What I should do is take a picture so that I can record the results on.
Sounds great.
Benchling actually.
I can shoot you over some files too, I assume, or is that unnecessary?
I mean, this stuff is. There's probably going to be a decent amount of data here. Like, let's try to work on getting the sort of base calling and the data processing into an AWS instance.
Sounds great.
Because then we don't have to eat up the hard drives on our computers locally storing all this sequence data.
I hear you there. Yeah, actually, AWS through TechSoup, we get like 2,000.
Yes, that seems very good. I wonder. Yeah, I do wonder how much computing on this will cost.
We wouldn't even need to do the computing, so to speak. I mean, I'd prefer to, but even if we just you're talking like secondary stuff.
Yeah, the secondary stuff. So
yeah.
Because you could imagine. I mean, you could imagine like not even bothering with base calling on the laptop because it's going to be slow. Like right now it's 24% of the way done base calling the just 55 megabases of data, and if we run it for significantly longer, that gap's only going to increase, and so I would imagine there's probably value in just sequencing stuff, not bothering with the base calling, uploading the raw sequence data in the FAST5 files to the AWS instance, and running the base calling on there, as long as it's not too expensive, because then you can get your base calling done like fast.
Yeah, I doubt it would be an issue whatsoever. Can we parallelize it as well?
Oh yeah, absolutely.
Awesome.
And depending on the base caller we download, so they have different. So all the base callers are neural networks, and they're constantly iterating on them to like improve accuracy and also improve speed. And so like the one that's in development now is called a Guppy, and it's optimized for running on graphics cards.
Oh,
And
salad.
that runs on the minute. The little thing that little device that you can plug your MinION into that will live base call for you, and will keep up with it. But yes, the Guppy base caller is much faster than Albacore, which is the one that is the current release one. But I think Guppy is not fully complete with the development. Although I could be wrong about that. I think maybe it's just that Albacore is the one they released to the public because they don't expect the public to have like random graphics cards they can port stuff onto. But like for their actual devices, you know, like the GridION, the PromethION, they actually have like those do like real-time base calling because they have graphics cards installed on them and like are running Guppy to base call the reads.
Yeah, we'll pimp some things out there, get it all working out well.
Sounds good. Yeah, if we can set it up so that it's as cheap as possible for anyone to do, that's the ideal. So that's why, I mean, like we could get ourselves a minute and it would be like super convenient. But I think there's a lot of value in figuring out like how to spin up an AWS instance to do that instead, and figuring out how much it costs per.
Yeah, it really just depends on what you want out of it. There's like thousands of not thousands, but there's probably like hundreds of combinations, like the way you want it set up. So it should be fine either way.
Cool. All right, that's been 10 minutes. Now let's add 500 microliters of the storage buffer. And the storage buffer, you will notice, is bright yellow. And there was more of it in this tube than I thought there was. I'm not entirely sure why they make these tubes opaque. It's not. That's actually quite annoying. I think this is actually fluorescein, based on the color. I could be wrong. But yes, bright yellow. And we're going to add 500 microliters of it to the MinION. Just suck a little bit up there and then squeeze it all down. Wash. Cool. So that's good for storage. Close the priming port. Close it. Close it up. And then, do we have a liquid waste container?
You can create one.
Okay. Yeah, because we want to, now we want to get rid of the liquid that's in here.
Any recommendations?
I mean, honestly, just like a glass. Yeah, one of those will work. Okay, cool.
We'll get this sorted out sooner rather than later.
All right. And you just get rid of the liquid waste, like so. Let's see, there's a waste port right here. And you suck it all up. Suck it up. Get rid of it. And let's do one more. Snaky snake. All right. That's
How effective would you say that is?
Hmm?
How effective is that? Like, is there any trial and error that goes into it, or like, can someone like me pretty much do that without
sucking this up?
Yeah.
Yeah, you can do it. Just make sure that the priming port and the spot-on port are closed.
Gotcha.
So that you can't suck air through them. Yeah, because like there's two holes right here.
Mhm.
So the idea is you can pull liquid through this, and then air will come in that way and allow you to suck it out.
Gotcha.
Yeah. So yeah, we've got our thing more or less fixed. I think are there a couple of bubbles on there? There might be a couple of bubbles on our MinION flow cell now. It wasn't super the most tidy, but oh well. Oh well. What's a couple bubbles between friends? And with that, we are done. And thank you, Anoop, for tuning in. And hopefully we'll edit this down to take out all the embarrassing and boring bits. Bye, everyone.